All right, so protein synthesis is a two-part process, so two stages. The first stage is called transcription, and the second stage is called translation. So make sure that you know the order. So you have transcription followed by translation. All right. Now, in order for us to make those proteins that we need, DNA has to be converted into a new language, and that new language is called RNA. And like DNA, RNA is a nucleic acid. And this process of taking the DNA, converting it to RNA, and then making the protein happens in two locations. So think about it like this. There are two steps, so there will be two locations. So for transcription, that first step, transcription takes the DNA and converts it into the RNA language, specifically messenger RNA. That's happening inside the nucleus. So that's the where. And then once the RNA is made, specifically the messenger RNA, it leaves the nucleus and it goes to the ribosome, and that's where the protein is going to be made during the process of translation. So key takeaways from this screen are that proteins are made in two steps. The first step is called transcription. The second step is called translation. Transcription takes DNA and converts it into a different language, RNA, which is able to actually leave the nucleus. And the RNA is going to then be read in the ribosome to make the protein during the process of translation. So as we move through this PowerPoint, make sure that you write down things as I point them out. So I want you to make sure that you know this next screen really well. So take a minute and write this down. Pause the video so that you have plenty of time to write it down before I start to explain. Okay, so hopefully you've written this down now. And how do we interpret this diagram? So first, let's look at the flow of information. So DNA flows to RNA to protein. So information goes from DNA to RNA to protein. How does it get from DNA to RNA? Well, that first step, remember, is called transcription. So in transcription, DNA is converted into RNA. Now the second step is translation, and that's when the RNA is read to make the protein. Now, underneath those arrows, you see where the processes happen. So transcription is happening in the nucleus, translation is happening in the ribosome. And remember, if you get confused about which process happens first, remember they occur alphabetically. You transcribe before you translate. So make sure that you know your flow of information. DNA goes to RNA to protein. How does it do that? Step one is transcription. What happens in transcription? Messenger RNA is made. Where does transcription happen? It happens in the nucleus. Okay. Step two of this process is called translation. What's made in translation? The protein. Okay. Where does translation happen? The ribosome. So again, make sure this is written down. You learn this slide really, really well. Okay, so before we go into the details of each step, I want us to take a minute and actually go through the differences between DNA and RNA and the different forms of RNA that you're going to hear me talking about as we go through transcription and translation. All right, so first let's just kind of look at RNA. So in general, RNA is a nucleic acid just like DNA is. However, it has a different sugar, so the beginning part of its name is different. So RNA stands for ribonucleic acid because its 5-carbon sugar that makes up its nucleotide is called ribose. Like DNA, it's made up of nucleotides. You might want to put here next to things that are similar to DNA, a letter S, so that you know that, hey, this is like DNA. So how is RNA like DNA? It contains nucleotides. Now, these nucleotides that are found in the DNA molecule, or excuse me, the RNA molecule, have the same structure as the DNA nucleotides. They still have a 5-carbon sugar, but now the 5-carbon sugar has a different name. It's called ribose. Right. It still has that phosphate group, and it still has nitrogenous bases. Okay, so first I'm going to do the things that are the same. So it has a phosphate group. That's the same. It has nitrogenous bases. That's the same. All right, it has a 5-carbon sugar, so that part's the same, but the name of the sugar is different. The sugar is called ribose. So that's a difference between the two of them. Now let's take a look at the nitrogenous bases a little bit closer. So if you look at your nitrogenous bases, RNA contains the nitrogenous base adenine, guanine, and cytosine, just like with DNA. So remember the S's that I'm putting on here stand for similarities between DNA and RNA. So those things are similar. Here's where they differ. Remember, DNA has the 5-carbon sugar deoxyribose, and it has the nitrogenous base thymine. Right. Notice there's no thymine or T. Instead, we have uracil. So uracil replaces thymine. Right, so that's going to be a difference between DNA and RNA. Another difference between DNA and RNA is that RNA is a single-stranded molecule, whereas DNA was a double helix. So that's going to be another difference. So DNA has 2 strands that are twisted on each other into the double helix, and the RNA molecule is a single strand, and that single strand can take on different forms, as we'll see in just a moment. So make sure you know, how are DNA and RNA similar and how are they different? So I tried to highlight that on this slide, emphasize it. So remember, the S stands for things that are similar, the D stand for things that are different. So now let's take a look at the different forms of RNA. So there are 3 different forms of RNA that you need to be familiar with. The first form of RNA that we're going to talk about is called messenger RNA, or mRNA. So make sure that you write down the 3 types. So the first type was messenger RNA, so mRNA. And notice that the messenger RNA has the instructions for making the protein. So these are the instructions for making the protein. And when I say instructions for making the protein, it contains the information that codes for the amino acids that will build the protein. So here are the instructions. How do we build that protein that we need to express that gene? You also have to know the pictures for these molecules. I want you to notice that messenger RNA is a linear molecule. Right? So it may look a little wavy, but it's a line. Right? And think of it kind of like a sentence. And these little shapes here represent the letters or the nitrogenous bases. And we figure out what amino acid we need by reading the sequence of those nitrogenous bases. So make sure you know messenger RNA, what does it have? It has the instructions to build the protein. And make sure that you can recognize its picture. It is a linear sequence of nitrogenous bases that will ultimately code for the protein. All right, our next type of RNA that's needed for the process of protein synthesis is transfer RNA, or tRNA. So I'll use the abbreviations most of the time when I talk about them. So transfer RNA, or tRNA, its job is to transfer amino acids from the cytoplasm into the ribosome. So remember, proteins are made up of amino acids. Those are its building blocks, or monomers. And those amino acids are coded for in that messenger RNA. So the tRNA's job is to go and get the amino acid that is needed at that moment to make up the protein sequence, or amino acid sequence of the protein. Now I want you to notice something about tRNA. Structurally, it kind of looks like a lowercase t. It's a little lopsided, but it still kind of has that basic structure. And here where this little open space is, that's where you're going to find your amino acid attached to the tRNA molecule. So our third type of RNA that's needed for protein synthesis is ribosomal RNA, or rRNA. So do word associations to help you remember what things go with or stand for. So think R, and that rRNA stands for ribosome. So it's going to make up the ribosome, so it's a component of or part of the ribosome. And the other thing to remember about that R, think of it as reading. So the ribosomal RNA actually reads the instructions. So all 3 of those RNAs work together to ultimately transcribe and translate a piece of DNA into a protein to allow for the expression of a trait. So make sure you know mRNA is the message. It contains the instructions to build the protein. tRNA actually carries the amino acids to the ribosome when they're needed. And the ribosome RNA or rRNA is actually reading the message, so it can tell the tRNA what amino acid is needed next. Right, so now that we've talked about those different types of RNA, let's take a look at the actual process of protein synthesis step by step. So remember, protein synthesis is a two-step process, and it's happening in two different locations of the cell. So this circle here represents the whole cell. Inside of there, this circle represents the nucleus. Remember, DNA is inside the nucleus, and it's a double-stranded molecule in the shape of a helix. So this is the double helix. Now, in transcription, only a segment of that DNA needs to be read, and a segment of DNA is called a gene. So only the gene that's needed for that specific protein that the cell needs at that moment will be read during transcription. So we're going to take that piece of DNA, the cell opens it up, and notice instead of like with replication where it copied both sides, it's only going to read one side of the DNA molecule, and it's going to make a single-stranded RNA, specifically messenger RNA, that will then leave the nucleus through one of these openings called a nuclear pore, and they are found within the nuclear membrane. Now that single-stranded RNA molecule leaves the nucleus and it heads to the ribosome, which is this light gray structure here. And remember, the ribosome contains a specific type of RNA called ribosomal RNA that reads the messenger RNA or mRNA. Notice it's reading at 3 bases at a time. Those 3 bases correspond to a tRNA molecule that has the amino acid that the cell needs at that moment. So the tRNA molecule brings in the amino acid, which are these little circles here. The amino acids connect together to form what's called a polypeptide chain, and that polypeptide chain will ultimately become the protein. All right, so now let's take a look at transcription very closely. So you need to make sure you know where it happens. So where it happens is the nucleus. What is made? All right, it's making mRNA. Okay, so mRNA is produced. And essentially, we just describe translation as the process in which DNA is converted into messenger RNA. So what happens in transcription? DNA is converted to messenger RNA. Where does it happen? It's happening in the nucleus, and ultimately it's producing the mRNA. And that mRNA leaves the nucleus and it heads to the ribosome. Right. Now, we're actually going to practice this process of transcription. It's very similar to DNA base pairing like we did in a previous lesson. So we're going to follow some kind of base pairing rules, but they're a little different when we go from DNA to RNA. So make sure that you write down this example as I go over it, and write down the little chart that I have on the next slide. So first, pause your video, copy down the slide. You need to make sure that you know what DNA codes for. So if there's guanine in DNA, what does it code for in RNA? If there's cytosine in DNA, what does it code for in RNA? And so forth. So pause the video, copy down the chart and the example, and then when you're ready, resume the video. All right, so if you're looking at this chart, we've got DNA and then RNA. So in the DNA strand that's being read during transcription, if there's a G, it codes for a C, or guanine codes for cytosine. If there's cytosine, it codes for guanine. Notice that hasn't changed. That's just like with DNA. G goes with C, and so forth. If there's a T, or thymine in DNA, that still codes for A, or adenine, in the RNA molecule. Here's where it differs, right here. Remember, DNA has adenine and thymine. RNA does not contain thymine at all. So when there's an A in DNA, it now codes for U, uracil. So the U replaces the T. All right, so now let's practice that. So notice how I have the DNA split up for you. The DNA is split up into 3 base segments so that it's easier for us to practice this process, because remember, everything is read 3 bases at a time. So here's your DNA. Now let's make the RNA that would pair with it. T would pair with A would pair with U, C would pair with G. And if you get lost, just go back up here to your chart and see what does it code for. G pairs with C, G pairs with C, C pairs with G. Okay. In our last sequence of DNA, T pairs with A, T pairs with A, C pairs with G. So that's how you transcribe. Very similar to pairing up two strands of DNA using the complementary base rules. So let's try another example. You don't necessarily have to recopy the chart, but I do want you to pause the video and copy down the example problem. All right, so here's our new example. So pause the video, copy down your sequence, TAC, AAG, CGA. Go ahead and try to do the DNA to RNA on your own and then resume the video. Okay, so hopefully you figured this out on your own and you've used your base pairing rules from DNA to RNA to get your sequence. So check your work. T pairs with A pairs with U, C pairs with G, A pairs with U, A pairs with U, G pairs with C, C pairs with G, G pairs with C, A pairs with U. All right. So that's what you should have for your second practice sequence. Okay. If you're not sure about how to do this, I will be hosting a live session to go over it with you. Okay. Moving on, now let's go on to step 2. So step 2 in the process of making a protein is translation. Now, translation, remember, it happens in a different location of the cell because the RNA leaves and it goes to the ribosome. Because remember, ribosomes are where proteins are made. So make sure you know, where does translation happen? It's happening in the ribosome. Now, what happens is a little more involved. So remember, the rRNA molecule, which is ribosomal RNA, its job is to read the messenger RNA. And that's what's happening in translation. Messenger RNA is read 3 bases at a time. You need to make sure that you know that 3 bases of messenger RNA are called a codon. And a codon codes for an amino acid. So that's a signal to the tRNA molecule, hey, I need this amino acid. Okay, so remember, the ribosomal RNA reads it 3 bases at a time. Those 3 bases on the mRNA are called codons. So that's the first thing that's happening in translation. The messenger RNA is read 3 bases at a time. Those 3 bases are called codons. Now, remember, in the diagram that I kind of went over with you, I talked just a little bit about how the tRNA molecule could pair up with those 3 bases that were being read. So the tRNA attaches to the mRNA at those 3 bases. And on the tRNA molecules, those 3 bases that are complementary to the mRNA that allows them to lock in place inside the ribosome, those are called anticodons. So make sure you know that we have codons on mRNA, we have anticodons on tRNA. And how I remember it is that I use little word associations or letter associations in this situation. So we've got this T in anti, and we have a T here in transfer RNA. And that helps me remember that the anticodons are the 3 bases on the tRNA that pair up or are complementary to the mRNA. And they latch together inside the ribosome so that the amino acid can be put onto the growing polypeptide chain of the protein. So as this process of translation is happening, you need to understand that it happens over and over and over again. Read 3 bases, slide down. Read 3 bases, slide down. And every time that the tRNA and mRNA connect, it's basically locking an amino acid in place, and this repeats over and over and over and over again. So make sure that you know from this slide that when the anticodon and the codon match up, locks that amino acid into place, and those amino acids are connected to each other through something called a peptide bond, and it forms a polypeptide. And this is repeated until it's completely formed. And that polypeptide, when it's completely formed, is called a protein. So you might want to put protein here beside polypeptide, so you know that polypeptide is just a fancy way to say the sequence of amino acids in a protein. Okay. Now we're going to actually practice translating the RNA into the amino acid sequence of the protein. And in order to do this, we have to use a codon chart. Now, I have placed in resources, or in your folder for you, 2 resources. There's a box chart and there's a circle chart. You can choose which one you would like to use. I personally prefer the circle chart, but either chart's fine to use. Okay. So if you go back and look at your examples that we did with transcription, we made 2 different sequences of RNA. I've written the first sequence of RNA that we made down on this chart. So you might want to just copy down that AUG, CCG, AAC that you see at the bottom. Notice there are 3 base segments. These are mRNAs. They would be called codons. So the codon charts allow us to figure out what amino acids are actually coded for. I personally would not have wanted to figure all this stuff out because there's 64 amino acids and there are 20 different proteins, so some of the words are repeat. And but this was figured out by Watson, I believe it was. No, excuse me, Crick, of the Watson and Crick group that kind of helped figure out the structure of DNA. He also was involved in kind of figuring out how the DNA codes for the proteins, along with some other scientists, but he did some research along these lines. All right, so let's take a look at how do we actually read this chart. So we read them 3 bases at a time, so that's a codon. So our first codon is AUG. All right, so how do I read the chart? Find your first letter, which is A. Look on the left-hand side of your chart. All right, find the letter A. So you have U, C, A, G. We found our letter A because it's our first letter. That letter A tells me which row I'm on. So I have to be in this row. I cannot move above or below it. I'm stuck here in this row in one of these 4 boxes. Now the second letter, notice up at the top of the chart where it says second base, that's where we look to see which one of the 4 boxes we should be in. So our second letter is U. So I'm on this row and I should be in the U box of this row. So I'm in the AU box. Now I cannot move out of this box. I can only choose from one of these 4. So how do I know which one I actually need? So look over on the right hand side of your chart. Notice where it says third base. All right, so my third letter is G. So I need to find AUG. So here I am. This is the AU box. This is the G line. So my first amino acid is methionine, which is abbreviated as MET. You just need to write down MET. You don't have to learn the full names. Now I'm ready to read the second codon. The second codon is CCG. So C is the first letter. So this is my first letter. I have to stay here in this row in one of these 4 boxes. I cannot leave this row. I cannot move up or down. So I'm stuck here in one of these 4 boxes. My second letter is C. So I need to look up at the top of the chart to see where the C box would be. So this is CU, this is CC. So I'm here in this box. I cannot leave this box now. I only can choose from one of these 4. And my third letter is G. So I'm down here on this line. Okay. So my second amino acid is PRO, which stands for proline. And if you notice something, sometimes everything within a box is the same because there are only 20 amino acids but 64 ways to express them. All right. So then we have AAC. So here's A. I'm on this row. I cannot leave this row. So I'm in one of these 4 boxes. So I'm in the AA box. So for me to find the AA, I have to look up at the top for the second one. So I'm going to be here. So I'm in this box, it has to be one of these 4. So I need the C line. So I'm going to look at over on the right hand side to see where the C line is. C in this box codes for ASN. So AAC codes for ASN, which I believe is the abbreviation for asparagine. All right, so now let's try that again. So if you didn't like the box chart, I'm going to do the second example using the wheel chart or the circle chart. So I'm a circle chart person, but both are fine and you'll still get the right answer whichever one you use. All right, so here's the circle chart. So here's our second sequence of RNA that we made earlier on when we were practicing transcription. Pause the video, write it down. So you got AUG, UUC, GCU. Okay, so that was the second one that we made. So the way that you read the circle chart is from the inside out. So A is our first letter. So I find it in the middle. Notice that this circle chart is divided into quadrants. Once I find that A, I cannot cross either of these lines. So I'm stuck here in this quadrant. So then I would just work from the inside out. So I go A, U, G, and then the outer ring is going to tell you the name of the amino acid. So that amino acid is MET. Our next one is UUC. So we go UUC. Again, just reading it from the inside out. So UUC codes for PHE. And remember, the outer ring has the abbreviation for the amino acid, which that one is called phenylalanine. And then we have GCU. So we go GCU, reading from the inside out. So GCU, that codes for ALA. And I would write that down. So those are our 3 amino acids that are coded for by those mRNA codons. And again, you can use whichever chart you prefer. And